Dr. Morgan Dendunker, a postdoctoral researcher at UCO. Morgan's work focuses on root trait diversity and the implications for forest function. Her presentation is titled Intra-specific variation of root traits in temperate tree species. Let's welcome Morgan.
Do you have a hand microphone?
I do not, but we could get you one. The hand microphone.
I would prefer it, but
Do we have that?
do you have one? Okay, perfect.
I don't, but it's
Perfect. Thank you so much. Okay. So it's okay? Yeah, everyone hear me? Okay, thank you very much for having me. So when you came here to the AGM this morning, maybe some of you were thinking about getting a coffee to get through the day. Maybe some of you noticed the beautiful colors on the leaves, but I'm pretty sure that nobody was wondering about what was happening on the ground, because roots are almost invisible. They are difficult to access, therefore they are difficult to study. Yet, it is a crucial part for plant functioning and ecosystem functioning. I'm not a root expert, sorry to disappoint you. And my goal here today is not to teach you all about roots, but today what I wanted to show you is how crucial it is to take into account root characteristics and their variation into ecological comparative study and ecological modeling. So you know that following a disturbance, forest will enter into a recovery phase during which recruitment and growth dominates. And it is really important to understand the characteristic of this regeneration phase to understand if we will have shifts in composition and structure in forests. So species will not react the same to a disturbance, and the species response to a particular disturbance will depend on functional traits. One of you wondered what was a functional trait this morning. So a functional trait is a measurable characteristic. It can be morphological, physiological, or phenological, and these characteristics influence how plant growth, survive, and reproduce. So traits define how plant responds to disturbances and how plants influence ecosystem functions. Most of the time, species present different traits value. So like in this graph, you can see that there is species 1A, species B. They present different trait value because they have different strategies to grow, to reproduce, to survive. The problem is we also have, within a species, we also have intra-specific variation. So rather than having one unique trait value per species, you will have this kind of distribution curve. So the range, the width of the curve represents the intra-specific variation. Why is there intra-specific variation? First, because plants are capable to adapt to their environment. So they show plasticity across environmental gradients. For instance, leaf shape, leaf area will vary across light conditions. The second thing that you probably know, because most of you are foresters, is that juveniles do not present the same characteristics than mature trees. That's what we call ontogenical effects. Third, there might be some local adaptation. So that is, it's a particular population that is really adapted to a given environment, and that will allow to perform better in a certain conditions. So this intraspecific variation has major implication for comparative ecology and for ecological modeling. Because of the traits are the link between plants and ecosystem functioning, so we tend to use traits in two models. And because traits takes time to measure, we often reuse existing trait data. The problem is, by reusing this trait data, when we extracted a value from a database, you don't know if this value corresponds to the mean of the species or if it corresponds to an extreme value, and there might be a value from a local adaptation. So the value that you extract from the database will not cover the context that the model is supposed to represent. And you don't have these distribution curves. We don't know them. So this has an impact on the model outputs, because using robust trait data as inputs will allow to have good outputs. It's like cooking. If you take bad ingredient to bake a cake, the cake will not likely, it will not taste good. So roots are key to understand several functions like biogeochemical cycle, salt structure, and that is why it is important to integrate them into models. But the problem is that the availability of root traits, they're not so great. Compared to leaf, for instance, you can see that for leaf traits, we have plenty of data. But for root traits, we don't have a lot of data. And even when we do have data, it's often categorical. For instance, we will know if a species has a taproot, or we will know if a species has more superficial roots, but we will not know the actual rooting depth. So this is why we launched Fun Roots campaign, which is, we wanted to build a comparative ecological study to really understand root traits variability. What we wanted to do is, first, we wanted to characterize the inter and the intra-specific variation in some root traits that are associated with drought sensitivity, because we know that drought is one of the major constraints for regeneration. The second thing that we wanted to do is we wanted to improve our understanding of seedling drought sensitivity using traits and trait data. Further down the road, that means that the outcome of this project will be used into species sensitivity indices, like Isabelle presented you this morning, but that's further down the road. And also, we wanted to inform about the use of trait data into models. So we had 3 research questions. The first one, we wanted to know how large is the intra-specific variation compared to the inter-specific variation. So in other words, do we have like overlapping distribution curves, like in the left graph, or do we have like distinct distribution curves, and that means that the intra-specific variation is lower than the inter-specific variation? Second question that we wanted to tackle is, if we take the total amount of variation for one trait, there is a part of this variation that can be associated with the inter-specific variation, and there is a part that can be associated with intra-specific variation. We wanted to know how much was associated between the two components. Third, we wanted to know what are the drivers of the trait variation, so the overall trait variation, and at what scales do they operate. If there are more local factors or more regional factors, we wanted to know at which scale do they operate. So we sampled in 3 regions across Quebec and Ontario along a precipitation gradients. We focused on 7 temperate tree species, and in each regions we had two light conditions. So we had closed site and we had open sites to have like contrasting condition, and we sampled 10 seedlings per species in each light condition and in each region. So that means that on the field we dig up like 340 seedlings. It doesn't seem much, but it's a lot of work because we dig up their entire root system with our hands and with nails. So lab processing involved like determine seedlings age, We also did some leaf area measurements, and at the end, we washed the roots, and we had like these beautiful pictures, really clear picture of the complete root system on scans. And what we did on these pictures is we measured some root traits that we know that are associated with growth sensitivity. For instance, maximum rooting depth, it's pretty simple. The more deep a root is, the more deep the plant will be able to get water. So the plant with deeper roots will be less sensible to water deficit than the plant that has superficial roots. A second trait that we studied is root branching density. Root branching density is like, it represents how much the roots is ramified. So it has to do with the volume of soil that the plant is able to explore. So we plotted the distribution of traits value for several traits. And what we have is these beautiful curves. And what we see is that the width of the curves, they are pretty wide, actually. So we have a high intra-specific variation. But we can also see that the mean of different species are also different. So that means that we have a high interspecific variation, but we also have a high intraspecific variation. If we look at this intraspecific variation and we look at it across traits, that's what we did by computing the coefficient of variation, we have traits that present high intraspecific variation, so with a coefficient of variation of like 40 to 50%. That's the traits up on the graph. And we have root traits that present very little, very small intraspecific variation with a coefficient of like 10 to 20%. Finally, we did a variance partitioning to see how much the traits variation could be attributed to interspecific variation and intraspecific variation at different scales, so at the plot scale, at the region scale. And what we see is that for some traits, we have like the green area dominates. The green area represents the interspecific variation. So this is good because if we have that, that means that the species are really distinct from each other. But on the top of the graph, you also have a dominance of red area. And red area means that the intraspecific variation at the micro scale, so within the site, at the seedling scale, these are factors that determine the intraspecific variation. So we have species identity and environmental condition that influence trait variation. So what does that tell us? For reusing these root traits, but that could be any traits from existing database, we can do the exercise of classifying the traits that we measured according to whether they are easy to measure or they are difficult to measure. And we are classifying them according to the extent of intraspecific variation. So first, for the traits that are easy to measure, I would recommend to measure them in situ, because it's always good to have the actual value. They are easy to measure, so let's go measure it. For traits that are more difficult to measure, but that present a low intraspecific variation, well, we could say, okay, it's okay to reuse them from databases because the intraspecific variation is low, so the value that we extract from database is likely to be good. Finally, the third category is a bit more tricky because these are traits that are difficult to measure, but that also have a high intraspecific variation. So for these traits, we might need further studies to find proxy that could be used instead of the actual traits into comparative ecology or into ecological modeling. Of course, making this classification and making these recommendations, there are several underlying assumptions, which there are three of them. First, we need to know the relationship between the traits and the function. It's often the case, but sometimes it's not the case. Second, the trait selection, the traits that you put into the study or the models, the choice of traits should be theory driven. You don't want to take a trait just because the data are available. If there is no mechanistic justification for using that trait, don't use it. Finally, if we want to study species as seedling survival, traits should correlate with performance metrics like growth or survival. So within diverse and within the team 1, Isabelle presented you the species sensitivity indices that we wanted to develop. So for sensitivity, we can define a species strategy into avoidance, resistance, and recovery. And up till now, the indices mostly taken into account leaf traits because data were available. So we had one ecological mechanisms. It's a shortcut, but mostly it was one ecological mechanism that was included. And with our study, we are able to include another ecological mechanism into species sensitivity indices. So that means it's a small step, but by doing this kind of study, we are building more robust indices, robust sensitivity indices that will be used into vulnerability assessment. So hopefully into a more robust vulnerability assessment. Thank you.
Any questions? Oh, we got two right there.
The point you made about not choosing all the traits, but leaving some behind. Is that somewhat subjective in the traits that you're choosing? In that, like, if you look, can't you find a function for everything? I don't know if you just comment on that.
It can be subjective, and if indeed you can find a function for every trait, but it depends on your study and on what you want to study. If you want to study drought sensitivity, don't use specific leaf area because there is no direct link between specific leaf area and drought sensitivity. I mean, it really depends on the context. So for each context, you will have a set of traits that will be different from another studies.
We had one more question over here.
What? Sorry.
Can we go back to the slide with the variance partitioning? I'm just curious.
Oh, sorry, I did the same mistakes then.
I'm by no means a forest ecologist. I'm just curious if you were to add like hyperlocal environmental variables to this, how much of it do you think would be
decrease or
explained by just, I don't know, like water table depth or things like that?
Well, that would be into the micro scale factors.
Yeah.
So I think they're already taken into account here. But what we could do is determine the actual relationship between water depth and intraspecific variation. But here it's like really a variance partitioning to see the different scales of the drivers. We are not looking at specific drivers, but rather at which scale do the driver operate. And we can see that the regional scale factors and the plot scale factors. So regional factors would be like the climates. And they are not influencing very much the intraspecific variation, which is a bit odd because most of the studies find a link between climates and intraspecific variation.
Good. Perfect.